Start. So it seems like a good time to start the defense. It's 10:02. And it brings me great pleasure to introduce Atticus Murphy, who is presenting his PhD defense public seminar. Atticus went to high school right here in Medford, and then he decided he wanted to travel away and did his undergraduate at University of Toronto. But even sometime during that period, he got lured back to Tufts to work with Sarah Lewis and Avalon Owens for a summer. And he decided to go immediately from Toronto, essentially back to being at Tufts again. And it's been really a delight for me to have him in the lab group. I think, you know, his knowledge of anything is encyclopedic. You know, he has a wonderful personality that I think kept a lot of us happy and going during all the shutdown years of COVID. And yeah, it's really been a delight. I think to introduce the topic of Atticus's defense, I remember strongly, discreetly, a day in 2016 when before Atticus was here, when the vice provost of research at Tufts sent this email to all the faculty saying that the MacArthur Foundation was going to give $100 million awards to people with great ideas to change the world, and faculty should submit an idea. And I was like, well, that's weird, and threw it aside. And then I was walking home through Somerville, and I thought, if I was going to make the world a better place, I would replace all these, you know, scrappy paved yards with flower gardens. And so I submitted that to the provost, and she was like, you don't work in this space. You don't know about this stuff. Why are you submitting this idea? And Atticus has been part of a cohort of students that included Nick Dorian here, Karen Dooley, Chloe isn't here, but is maybe online, and James Michelini, who all converted that vague thought I had one day walking home to a whole research program and a whole outreach program that astounds me with its amazingness. And Atticus, of everyone in that group, took on the topic that I knew least about, that was the most half-baked in my mind, which is bringing in human interactions to the urban ecology of spaces, and did wonderful things with it, which he will tell you about today. So, Atticus.
Hi, everyone. I'm Atticus, and I'm really excited today to get to talk to you about my dissertation, which I titled The Gardening Bug and Gardens for Bugs. I want to open with a question now, which is, what do these three things have in common? So first, the prescriptions of a newly created drug, recycling, and the design of people's front yards. And I was told I have to give you like 5 seconds to think about it, otherwise it's too fast. So think about it for 5 seconds. Okay, these behaviors are all socially contagious. And what does that mean? These are behaviors that arise because humans often mimic what the people with whom they interact are doing. They mimic the people they interact with the most. So in the case of these prescriptions of a newly created drug, doctors will start to prescribe the drug at higher rates when their friends who are also doctors start doing that. People will recycle more if they see their neighbors are recycling. And people tend to make the fronts of their houses look pretty similar to their neighbors. And just to explore that last point, just generally, yard landscaping seems to conform to pretty strong social norms. In the United States, the current social norm is this, unfortunately, pretty biodiversity poor thing, which is lawns, turf grass lawns, are a powerful norm. If you want to see a visualization of how powerful, this is the suburb on Long Island, Levittown, one of the original modern suburbs in the 1940s. That's what it looked like when it was first built. And if you go back there today, those streets look almost identical. That's how strong these social norms are. It's all turf grass and a little bit of hedging. However, there is a small but growing movement of planting diverse pollinator gardens. And my dissertation aims to test whether this novel practice might be socially contagious. In other words, could gardening be spreading between neighbors? So the origins of pollinator gardening are really the realization that urban flowers are valuable habitat, particularly for insects. And this has become really relevant because in recent years, ecologists have been really concerned that a lot of insects are in decline, and that includes pollinators. Pollinators are important for a lot of reasons, but one of the main reasons is that they visit flowers. They eat nectar and pollen at these flowers. And in doing so, they pollinate over 85% of all the flower species on Earth. So these are flower species that pretty much can't exist without insects. And urban gardens turn out to be home to pretty surprisingly diverse insect communities, making them kind of appealing to target for habitat creation. So pollinator gardening involves planting diverse flowers in order to feed these insects with nectar and pollen. One of the key facts I want to just introduce to you here is that in gardens, flower diversity and flower abundance are really closely related to pollinator diversity and pollinator abundance. And put simply, that means that more flowers and more types of flowers leads to more pollinators and more types of pollinators in that garden, right? And for that reason, my dissertation focuses on all types of diverse flower gardens. So that includes pollinator gardens in the strict sense, but also ornamental gardens, diverse vegetable gardens, anything that provides a diversity of flowers. Another context for my work is that urban lands are kind of this growing focus for conservationists and ecologists just generally. And pollinator gardens are part of this broader trend. One of the reasons for this is that globally, actually only in recent years this happened, globally a majority of people now live in cities or suburbs. And so most people's regular interactions with nature on Earth now happen in cities. The other realization is that cities have substantial total green space, maybe more than you might think. And residential areas, so places like this aerial photo, make up a really big portion of that green space. It's not just parks. For instance, in Boston, front and backyards make up 43% of the total area of Boston. So it's a really substantial type of land. Just to drive that point home, here's a map using remote sensing data from NASA. I didn't make this map, but the black is turf grass, right? So that's lawn. And if you look, that's a lot of black on this map. In fact, it's enough black that turf grass lawn would be the largest irrigated crop in the United States by area. And it would cover, if it were in one clump, the state of Georgia. So these are really substantial areas of land that we currently devote to turf grass, mostly. A lot of opportunity here, a lot of opportunity for habitat creation. But there's also some concerns. And one of the concerns is that urban habitat might be fragmented. And I'll explain what I mean by that. So the rationale for this concern is that individual yards are quite small. You can see on that graph on the top there, that's what an individual typical yard looks like. It's not huge. And so animals, in order to sustain their population, will need resources from many yards. If landowners start acting independently, this could create the risk of fragmentation, meaning that good habitat is not nearby other good habitat. And if animals need habitat across multiple yards and they're too far apart, then they can't live there. One set of solutions to this problem is to kind of try to coordinate management, to make people make the same decisions and then create habitat nearby other habitat. And this has been called like scaling up from gardens. But I think an underappreciated fact is that if contagion were to occur, this would provide a bottom-up mechanism for reducing fragmentation, regardless of top-down inputs. And the reason for that is that neighbors would be behaving similarly to each other, which would create self-organized clusters of gardens. And in ecological terms, that would make a more structurally connected landscape, where structurally connected means that habitat patches tend to be closer together. So on this cartoon, if we imagine that blue patches are gardens, on the left, we have a fragmented situation where managers are all doing their own thing. There's no coordination between them and no contagion, and the blue patches are pretty far apart. Whereas on the right, if there's contagion, then the gardens, the blue circles, tend to be closer together, right? They're all in a clump. And that's better for animals, like pollinators. So the outline for the rest of my talk is going to be that in chapter 1, we're going to talk about basic patterns and drivers of flower gardening, kind of just to make a map and get a sense of where flower gardens are. Then in chapter 2, we'll develop a test for social contagion of gardening, specifically accounting for that background variation in gardening. And then in chapter 3, we'll start to try to explore whether this matters for pollinators and explore those ecological impacts on pollinators of socially contagious gardening. Spoiler alert. But first, the basic trends, right? So pollinator gardening is what I would call participatory. And what that means is that in order to be effective as conservation, you need a lot of people. Like I said, you need many yards. And so kind of in response to that, there's been some past work describing motivations of who's gardening, right? And they actually have kind of a wide array of motivations. A lot of people care about beauty in their yards. A lot of people care about feeling like they're helping nature, etc. But there's less known about the current prevalence of gardens, like how many people are gardening as a proportion of the population, and also what factors predict whether you're gardening and the diversity within your yard, which I'll point out is the key factor here for pollinators, right? To support diverse pollinators, you need a diverse garden. So with that in mind, this chapter, I ask 3 basic questions. How much flower garden habitat is there? What factors might predict flower gardening? And what factors might predict diversity within those gardens? And specifically, I look at 2 factors here, income and lot size. And I'll explain why here. So income is often predictive of urban biodiversity. So this has been called a luxury effect. Rich people get to experience more diverse nature in cities. It's been found in many different taxa, many different types of organisms, including plants. And it's been linked to various things like increased ability to spend money on landscaping and plants. Maybe they have different landscaping preferences, whatever. But regardless of the reason, the expectation thus would be that flower garden and diversity should be linked to higher income areas. Lot sizes also could have a really important effect on landscaping and gardening behaviors. Lot size means that the area of the land that you own, and larger yards tend to have more different landscaping types. So in this image, we might say that one landscaping type is the turf grass and another type is the hedging. And if they added a garden, we might say that's another type. So you might think that small lots might have small yards, and this could be a constraint to gardening. There's not enough room to start a garden. There's also an expectation from basic ecology on the diversity front that smaller yards should just have fewer species in them. Small patches in nature tend to have fewer species than big patches. So this leads to our expectation that flower gardening and garden diversity should be linked to larger lot sizes. So high incomes and large lot sizes. That's our prediction. To address these questions, we needed a map, right? And we selected 11 cities in Greater Boston. These included dense cities like Somerville and Cambridge that look like the image on the left mostly, and also kind of medium density suburbs about 10 or 20 kilometers away that look like the image on the right. We selected these cities pretty intentionally because we wanted to decouple our 2 predictors, and that's why we only chose 2 predictors. Some studies throw all the predictors in a hat, but we wanted to a priori say we're decorrelating 2 correlated things. In the United States, big lots tend to be wealthier. So we needed to carefully select cities that independently vary in lot size and income. We also wanted to develop a method of doing large-scale mapping, which I'm going to use for the whole rest of the talk. And we used Google Street View. This provides 360-degree imagery on street level of people's the front of their houses. So this study is only about front yards. And what we did is we manually looked at all the images that we could. We pulled them from the Google Street View API, and we scored them as either being a flower garden, like this yard, which you might recognize. It's on the walk from Tufts to Davis Square, or as one of these 4 types of non-gardens, which we mostly used to help us distinguish between flower gardens and not flower gardens. So we said there was also lawn, weedy yards, shrubby yards, and non-vegetated yards. And I would love to give a huge shout out to Audrey Carver and Daisy Bonifant, who hand scored what you will see is a large amount of data as undergraduates at Tufts. When the data was collected, it looks like this. So this is a tip, the tip of North Cambridge is highlighted in gray here. All of those gray dots are scored addresses, and they are non-gardens of different types. The green dots are flower gardens. And our overall data set included every residence in the 11 cities, except for large apartment buildings, because they don't really have traditional yards. And that gave us 101,000 addresses total. We got that from the tax assessors. And then we fed that into Google, downloaded the imagery. We got 86% of those images were scoreable and we scored them. We supplemented that data, which was remotely gathered with our computers during COVID, actually, with some in-person surveys. So we picked 500 random front yards in that data set. And we stratified that across all the categories and cities so that we ended up with 100 yards of each type, 100 gardens, 100 lawns. And we did 3 visits during the summer to record how many flowering species were in bloom in that yard. And we categorized each species as either being cultivated, which are the intentionally planted species, or spontaneous, which is a technical term for a weed, which means that they are probably not intentionally planted. And for this whole talk, we're only going to talk about cultivated species. There were 2 things we wanted to use these in-person surveys for. One was to kind of ground truth our remotely gathered data. We wanted to see how diversity was different on the ground compared to our street view gathered data. And then we also wanted to address those questions that I mentioned about what drives within garden flower diversity. To do this, we used 2 different modeling approaches that were really similar. We used binomial mixed models, predicting the probability of flower gardening versus other lot types. So this is to get at the idea of how many people are gardening. We did this on a per census tract level. You could think of that like a neighborhood, and we had fixed effects of the median lot size and the median income of that neighborhood. We also had random effects of city, census tract, and block group. And this was intended to account for spatial autocorrelation, which I won't get into, but you can ask me about it if you care. For the within garden diversity, it's a similar approach, but here we're using a negative binomial mixed model, and that's because we're predicting species counts, so the number of species per yard. And that, of course, will be on a per yard level. Each yard had its own species richness of flowers, and we then have fixed effects of lot size and income again, and the same random effect structure. So to answer the first question, how much flower garden habitat is there? We scored a little over 86,000 street view yards. And that gave us data that looked like this if you put it in a colored bar chart. The green color there is lawn. And you see that lawn was indeed dominant in our study area. 58% of front yards were lawns. About 2% of all the front yards had flower gardens. That's the little pink bar up at the top. So what's going on the ground at those addresses using that 100 gardens that we visited and the 100 yards in each other category? So we saw that street view flower gardens had about 3 times the floral species richness per yard. That's the y-axis here is the mean species richness per yard. They had about 15 species per yard on average if they were a street view garden, which means a garden that we scored as a garden on street view. And all the other categories had something around 5 species per yard. And that led to this trend that the gardens as a whole, our 100 gardens, had an estimated total floral diversity of 300 species, which is really big considering the small size of our yards. And that was about double any of the other yard types. So lawns had a total richness of about 150. And just as a reminder, richness is the number of species of flowers. Next, we wanted to ask what factors predict flower gardening. And as a reminder here, we're looking at lot size and income on the y-axis. And we predicted that large lots and high incomes, so the top right of this graph, we predicted that that would have the most flower gardening, and that would be the deepest red color here. What we actually saw was that there were more flower gardens in neighborhoods with high incomes and small lots. So the top left of this graph, the dashed lines are the averages. So what factors predict diversity within gardens? And we test income and lot size again. So again, here we were predicting that income would also lead you to having more flower species within your garden, in addition to making you more likely to garden. And we saw a weak trend towards that, but a significant one. So there was a messy positive relationship between income and the species richness of flowers in your yards. And it actually applied, all these different colored lines are the other types of yard. And it was the same relationship across all the yards. There was no interaction, which was interesting. We expected that lot size would be positively related to floral species richness per yard. Bigger patches should have more species, right? But we actually found that there was no relationship between lot size and species richness within the yard for any of the yard types, which was really surprising. So what do we make of these kind of basic patterns of flower gardening in this chapter? So first, flower gardens are rare but important. So they were only 2% of all of the yards in our data set. But when they're there, they drive big increases in floral diversity, right? So there were 3 times as many species per yard, 2 times the total diversity, just as an illustration of what that leads to. 93 flower species were only ever seen in flower gardens, and the comparable number for lawns was 4. So flower gardens are contributing vastly more to floral diversity, like punching way above their weight. We also saw that income was linked to flower gardening and diversity, and this is consistent with a lot of past literature on the luxury effect. But I would also note that, like a lot of past literature on the luxury effect, there's other correlates of income, right? And even though we were able to decouple lot sizes from income, we didn't decouple education from income. It's too hard to decouple that many factors at once. So in our data, education was linked to both income and flower gardening. And so understanding the kind of the causal relationships between these things is a challenging future direction. Unexpectedly, we saw that small lots actually had more gardens. This is Elizabeth's old house in Somerville. You'll see she had a garden on a very small lot. And when we started reading, we started to think maybe this wasn't as surprising as we thought. So previous studies suggest that bigger yards have more turf grass and more trees as a proportion of the yard. They use more of their yard for turf grass. And it could be that lawns aren't really seen as worthwhile on really small urban lots. You don't want to buy a mower. What are you using the lawn for if it's a postage stamp? I think this could have some relevance for American urban planning in some interesting ways. Most of the country, at least most of the new parts of the country, operates with minimum lot size zoning policies, which mean that new developments have a minimum lot size that's actually quite large. And so it would be an illegal house to build. And it is also a house that is much more likely to have a flower garden in our region. And so I think it's really interesting to think about how these policies are affecting ecosystem services and biodiversity. So now that we understand the basic pattern of flower gardening, we can move forwards and try to develop a test for social contagion that accounts for those basic patterns. When we look at this data, what we're really talking about is why did these flower gardens get created where they were created? Each of these green dots is a garden. In the last chapter, we explored kind of one set of factors that are contributing. So characteristics of households and their yards, something like income of the household or the lot size. But at the same time, we want to test for a different set of factors, these social feedbacks between people that I want to emphasize are happening at the same time as they're happening, right? There's demographic correlates and there's also social feedbacks. And we need a way to distinguish those in order to ask these 2 questions. So first, do we see evidence for social contagion in flower gardening? And if yes, how does contagion affect connectivity of garden habitat? So when we're thinking about how to develop this test for contagion, we need to think about how contagion would produce localized garden clusters. And I'll explain what that means. Last chapter, we were exploring kind of neighborhood scale variation in gardens. So that was tract level data. We're predicting the proportion of all yards in a neighborhood that have gardens. Social contagion would operate at a much smaller scale than that. It would be strongest between nearby neighbors who interact more. They see each other's yards more, and they also may talk more. And that means that the key signal of contagion would be elevated frequencies of gardening, specifically among the nearest neighbors to gardens, over and above any factors in the neighborhood, like who lives there. We're going to use the same Street View data to answer this question. The way we do that is we zoom in on an address. Conceptually, this is how we do it. We zoom in on an address and we say, okay, there's 264 Lexington Street. Let's look at the nearest 10 neighbors and count up how many gardens there were. In this case, there's 0. We can do this for a set of random non-gardens and then the set of gardens, with the prediction that the set of gardens would have more other gardens around them. In this case, there's 3, because I specifically chose this to demonstrate the point. Our modeling approach here was, again, a binomial mixed model. And here we're using a fixed effect, so a predictor of the type of address. So you have a neighborhood that is either centered around a garden or centered around a non-garden. We did this for all of the gardens in our data, which was 1,940 of them. And then we picked 1,940 random non-gardens. And here we included random effects, again, of city, census tract, and census block group. And here the aim is to account for those larger scale neighborhood factors that are not contagion that also drive clusters of flower gardening, people living in certain places. So here's what the data looked like at one scale. So let's say we looked within 5 neighbors of a garden. On the x-axis, we have the center point. So are you near a garden or near a non-garden within 5 neighbors? And the y-axis is the proportion of all those yards that are estimated, the estimated proportion of all the yards that have gardens, right? So 1 would be 100% have gardens. It's not 100%, I can tell you. What we see is that there's twice as much gardening within 5 neighbors of a garden, which is neat. But this scale, 5 neighbors, was chosen arbitrarily, and we wanted to look at this across scales. And so we take this difference, what that orange arrow is showing, and we call it the effect size of the neighbor gardening. What's the boost to your probability of gardening because you're near a garden? And we look across scales. So on the x-axis now, we have the number of nearest neighbors, how far away is that garden? And on the y-axis, we have that effect size. And what we see is that gardens are much more common near other gardens, and furthermore, that the nearest neighbors have by far the biggest effects. In fact, nearest neighbors of gardens have a 3 times higher gardening rate compared to background. And this effect is significant up to 10 neighbors away, but it kind of tails off pretty quickly. And by the time you're out to 30 neighbors away, it's pretty much 0. Most of the action happens within about 5 neighbors. And I would point out that 5 neighbors is really not a big area. If we look at this map of Cambridge again, the tip of Cambridge, I've circled 5 neighbors there, and you can see pretty much all of those neighbors from your front door. And we think that's a key piece of evidence here for this being social contagion. We saw the strongest effect between next door neighbors, and that suggests it's not just driven by factors like you having a similar lot size or income to your neighbor, because then as you move further away, it drops away really quickly. In other words, it's probably a smaller scale than the variation in those other confounding influences. So we see this really localized clustering. But what does that mean for connectivity? So we tested this using simulations, and we reshuffled all of the flower gardens randomly across scales, which means we imagined a world in which the flower gardens were truly randomly distributed. And then we let them be randomly distributed by city, and then randomly distributed by census tract, and randomly distributed by block group, right? So we're looking across scales and allowing that reshuffling to happen. And we compare that to our observed data. And the aim of this is to let us distinguish clustering that's due to contagion versus clustering that's due to the demographics of neighborhoods, what we were talking about in chapter 1. And we measured connectivity with 3 things. First, we used Clark-Evans index, which I'm not going to show you, but I want you to know I did it. And then we used the average distance between gardens in meters, and the percent of gardens that are nearby to other gardens. And here we defined nearby as being within 50 meters, which is broadly consistent of estimates of the typical distance of the foraging distance of a typical solitary bee on a typical trip. And solitary bees are a really important group of pollinators in the city. And this actually is an estimate that is partially informed by experience from work in our lab, from Nick Dorian, Chloe, and Karen Dooley, who have all done projects with that beautiful green bee. So when we look across scales, we can see that on the y-axis here, we have the mean distance to the nearest garden, how far apart are gardens on average, and we can see that contagion increases connectivity, which means that it decreases the distance between gardens, the average distance. As we go from random to observed, we go from about 138 meters between gardens to about 120 meters. On the right now, we're looking at the percent of gardens that have another garden nearby that's within 50 meters. In the random situation, if we distribute those 1,940 gardens randomly, about 15% of them are nearby to each other. But in the observed situation, 30% of them are nearby to each other, about twice as many as random. What you'll see is that neighborhood factors like income and lot size can account for some of the clustering. When we cluster by city, tract, and block group, we go up a little bit in the percent of gardens that are nearby other gardens. But we actually observed much more clustering than that, up to 30% from 20% are nearby. And what do we conclude from that is that contagion contributes to the clustering of garden habitat. It shifts us some amount towards that connected situation on the right, which is probably good for animals. It reduces habitat fragmentation, which might reduce the need for top-down efforts to scale up habitat creation, which I would say are not a waste of time, but might be less worrisome if contagion is occurring. One of the things we don't really know is how does this clustering of habitat actually affect urban pollinators? We think contagion will lead to clustering, which will lead to connectivity, which is good, but we don't know that much about how this will affect pollinators. And I tried to start getting at that in my next chapter, which is about the ecological impacts of contagion. So some areas in our data have more gardens than others, and that's partially due to contagion. That means that there's more habitat in some neighborhoods compared to others. And we don't know how insects might be affected by this. In other words, what I'm going to be focused on is do insects use habitat in some neighborhoods more than others? We're focusing on just one species here, so we can't generalize to all pollinators. And it's the monarch butterfly, that beautiful orange butterfly that you probably can see in your yard throughout the summer. Monarchs lay eggs on milkweed. If I say host plant, that's the plant on which an insect lays eggs or reproduces. Caterpillars eat those leaves until they're ready to pupate, and then they become adults, and it happens again. They're common in urban gardens, but they're also a focus of conservation right now because they've declined a lot in recent years. And we're going to test how monarch use of host plants, milkweed, varies with urban neighborhood context in order to understand how the clustering of habitat, partially caused by contagion, might affect this pollinator. For butterflies, there's actually two types of resource. There's host plants on which eggs are laid, which the term is oviposition, so I might say that. And that's what the caterpillars eat when they're babies. And then there's nectar plants on which the adults feed. So the females are flying around laying eggs on host plants. And they're also, kind of fueling up at these little Gatorade stations and getting sugar water, which fuels their flights, lifespan, and reproduction. So we can think about these two resource contexts. If we imagine a single stem of milkweed, and we're going to measure how heavily this stem of milkweed is used. So we're going to put a circle around it. This is our focal plant, and we can vary the background density of host plant. How many other plants are around this plant? And that's a way of looking at habitat clustering, right? How much habitat is there around this plant? We would predict, this is just what we predicted a priori, that host plant density and per stem oviposition, how many eggs were laid per plant, would have this hump-shaped relationship. And this is a sketch because it's not real data, I just sketched it. At low densities, we would think that there would be reduced attractiveness. Maybe it's too hard to find those stems, they're really sparse. And so we might see lower per stem oviposition. We might see peak use at the kind of intermediate densities. And then at really high densities, they might get satiated. So they might kind of run out of eggs. There are so many stems of milkweed around, they don't have unlimited eggs. And so eventually there's so many stems that you see fewer eggs per stem in really, really dense patches. So we could see either higher rates of per plant use with increased host density or lower, depending on where on this curve we are, where on the x-axis our landscape is. I will tell you that previous work in natural systems has mostly found satiation and dilution in butterflies and in monarch butterflies. They run out of eggs. but we don't know how this holds for an urban context. We don't know where on this curve we would be. Remember, there's also nectar plant density is another factor, right? So again, we're going to focus on this cartoon on a single stem of milkweed, and we're going to monitor it for use by monarchs. So I'm putting a circle around it. That's our focal plant. And now we're varying the amount of nectar around that plant. You can think of nectar like gardens here, right? How many gardens are around this plant? Contagion would lead to more gardens being around this plant. Here are predictions about how oviposition rate or host use, how that would vary with nectar density are simpler because we just think more nectar means more host plant use. And that's because of a few reasons. I'll just give a few here. So monarchs that are feeding on more nectar get longer lifespans. They get to have more babies. And they also move slower. So the adult females spend a lot of time nectaring. If you ever watch a butterfly in your garden, if you have nectar, they're going to stick around a lot longer. So they're moving slower, and that might mean that the stems that are nearby nectar get more use. They get more eggs laid on them because the females are just hanging out in that neighborhood. And so we predict this sketch curve that with increasing nectar plant density in the neighborhood or increasing gardening in the neighborhood, perhaps, you might see greater habitat use, more use of host plant. We don't understand the shape clearly, but we're just saying it will go up. So how did we experimentally test this stuff? So we put out identical 2-foot wide pots of swamp milkweed, which is one of the host plants for this butterfly. And we did this in urban parks, which was really fun. We met so many community members. We had these little signs that I think Karen designed, and we had a QR code explaining what was going on. And the plants did really well. So they went in at about like 6 inches tall, and they ended up a meter tall. I don't know. I mixed my units because we were measuring them in centimeters by the end. So they were like a meter tall. They're half as tall as me. So they did great. There was 1 pot per park. And I say identical, like they had the same number of the same type of plant, right? And I just want to give a huge shout out to Karen Dooley, who is here today. And this is us at the end of our field season celebrating. Karen made a little newsletter telling everybody how many monarchs were in their park. This all wouldn't have been possible without her, both because she did so much of it, and also because she came up with a lot of these ideas when she was working on her undergraduate project the year before, and is now a co-author on this. So, hooray. We surveyed for monarchs by just turning over every leaf in the pot and looking for eggs. That's what a fresh egg looks like. It's this creamy little football-shaped thing. We did this every 3 or 4 days for the summer. And we then totaled up how many fresh eggs we got per pot, per site. So fresh would mean they were laid within the past 3 or 4 days. And we can then sum the number of eggs each pot got. And they're identical pots, right? So this is a way of measuring host use in that neighborhood. To characterize the neighborhood, we needed to measure host plant density and nectar plant density, right? So to do host plant density, we just walked around the streets in the neighborhood, and we counted how many milkweeds we saw. So, that yard had 135. That was probably the one that had the most, but that's why I picked it. You probably can't read the number though. And that was within 125 meters of our pot, which is that orange dot there, and that circle is 125 meters. So we walked all the streets that we could see, and we counted how much milkweed was in the neighborhood. To measure nectar density, we used gardens as a proxy. So we said, what's the frequency of gardening around this pot? And that percent is what we used as our proxy for nectar, because gardens, as I've said, they provide diverse nectar sources, and monarchs are commonly in gardens eating that nectar. So looking at some results. So monarch oviposition actually varied with both resources. We saw a trend towards satiation, so a negative effect of milkweed density, but mainly we saw a negative interaction, a significant interaction between milkweed and gardening. So let's look at what that looks like. So on the x-axis, we have neighborhood milkweed density, and on the y-axis, we have gardening rate. So host on the x and nectar on the y. The shade of orange on this plot will be the predicted number of eggs a stem would receive in that neighborhood. So that's like how much host use was there in that neighborhood. And the black points on this plot are where we had data. So I'll walk you through this step by step because interactions are always annoying to talk about. So you'll see that one of the things that jumps out at you is that at low milkweed densities in this purple box here, we see a positive effect of gardens. And that's as we predicted, right? There's more nectar in those neighborhoods, and that could be partially due to social contagion. And monarchs are using host plant in those neighborhoods more. And as you go up in gardening rate in this region of the plot, you get more eggs, darker orange. Kind of unexpectedly, this effect fades away and actually might become negative out in high milkweed density areas. This is not as we predicted, but you'll note that there's very few black points in this box, right? And here we're looking, going up this box, we get higher and higher gardening rates, and it actually gets less orange. Now I'm going to highlight in green some milkweed density effects. So at low gardening rates in this green box at the bottom, we see that higher milkweed densities might be attractive. It gets a little bit more orange as you go from left to right in that box, meaning that when there's very few gardens around, having more milkweed around might be attractive for butterflies. But again, that region of the plot doesn't have much data because those neighborhoods didn't exist in our region. Last, at high gardening rate, we see pretty good evidence for a negative effect of milkweed density. And this is satiation, which is what we predicted, right? So what this means is in garden-rich neighborhoods with lots of flowers around, more and more milkweed means less eggs per stem. They get satiated. And that's as we predicted. So what does this mean? So monarch host use varied with context. Overall, we saw evidence that monarchs get satiated at high milkweed densities, which is what people predict for natural systems too. There's so many stems, they don't use them all. But flower gardens also increased oviposition in low milkweed neighborhoods. So when there's not that much host around, and there's a lot of flowers around, potentially due to social contagion, we saw greater host use in those neighborhoods. It also might be that gardens were actually only a significant source of nectar. Remember, I said I was treating them like a proxy for nectar availability. It might be that gardens themselves were not an important source of nectar unless they were in the high end of what we observed, which I'll point out was only 8%. That's the maximum neighborhood level rate, right? And so, maybe we need a certain amount of gardens for them to actually make a difference for insects. However, in contrast to what I just said, monarchs also did lay eggs in every single neighborhood, which is pretty amazing. These are little containers, and we saw eggs across every single context. So to conclude, I will start with just summarizing what we found in each chapter. So when we looked at the patterns and basic drivers of flower gardening, we found that they're rare but important for floral diversity, and that they're linked to high incomes and small lots. We found evidence that flower gardening looks like it's socially contagious, and this leads to significantly improved connectivity of gardens. And then last, we found that there are some impacts of clustering of habitat on the landscape. It mattered for monarch habitat use, but all parts of the landscape were used, which is kind of encouraging if you want to plant gardens. Just to kind of reflect on all of this, urban ecology is a pretty new field. It's only really gained steam and a lot of people doing it in the past 30 years or so. And I think one of the things that I feel like I've learned is that interdisciplinary approaches are really valuable for understanding these processes. Because the social sciences give us these insights into how human decision-making can create or constrain urban habitat. All of the urban habitat in a city is under some kind of human influence. And then ecology provides the tools for quantifying these spatial processes, habitat functioning, and the impacts on animals that this ecosystem has. And I would just say that I think basic ecological theories that might have been developed for a totally different system, like a natural or agricultural system, can also be useful for understanding urban ecosystems. Specifically in kind of contagion ecology, I think there's like so many exciting questions that hopefully someday I can ask. Like, which people and which gardens are the most contagious? What qualities of those gardens are there? Can we seed neighborhoods with super spreaders by giving them the tools to become those contagious gardens? And how are pollinators affected by contagion? I tested one element of one pollinator. I don't know how other ones would be affected. I think I've gotten a lot of hope from this work. Like, gardens get used by insects. And if you add a garden anywhere, it'll get used by some cool insects. And then every garden that we add brings us closer to meeting habitat requirements for species that aren't yet in the city. And that's just really neat. And I think a specific finding from my work is that every new garden you add brings more non-gardens closer physically to seeing a garden every day. And I think that is in itself a great conservation impact because it makes this practice more contagious. I want to give a shout out on that second point to the Tufts Pollinator Initiative, who I just was so privileged to get to work with all of my friends on doing tons of outreach events, and planting gardens and giving plants away and helping people start their gardens. And it was a real privilege. And I think just overall, I have really enjoyed and think it's important to understand that this work is about direct connections between people and ecosystems. And I think it's really nice that it's not abstract, and it's these people making direct choices with their lives that affect an ecosystem. And with that, I would like to say thank you. I'm going to do my acknowledgements first and then take 5 or 6 minutes for questions if I have time. We might go 5 over, but we have the room for the half hour after. And you can leave if you want to. I first want to thank Elizabeth. Probably gonna cry. I was like so lost for 2 years in my dissertation, and it really meant a lot that she let me just start this random project that I had no idea if it was going to work, and she didn't either. And I was so unhappy with my monarchs in nature, and I found monarchs in the city, and I was like, okay, now I'm happy. And I found gardening, and it meant a lot to me that she mostly just wanted me to be happy, and I think that was just the best advising thing that could have been. I'd like to thank my committee, who's been incredibly helpful. Colin, Michael, Caleb, who brought a little bit of sociological sanity to the whole thing. Emily Miner, who is here as my external examiner, and also Sarah Lewis, who used to be on the committee but has retired and was really influential in the first 3 years of my dissertation. And also actually advised me in my rotation project and also as an undergraduate when I was working here in the summer. I'd like to thank the Crone Lab, so all the current people, James, Sylvie, Aramy, Lucy, Emily, Erin, Jesse, anybody I forgot, I'm sorry, I made this at literally 2:45 in the morning this morning. Also the past cronies, Nick, Brendan, Rachel, Nat, Jenny, Colin, Lee, Leslie. This is a picture of us from just this last month, and it's so fun to still get to see people at conferences. I'd like to thank all the undergrads who were totally instrumental in doing all this work. Karen, who I will say more about in a minute, Alexandra, Christina, those were both field technicians with me for summers, and then Daisy and Audrey gathered literally all of the data that this entire dissertation is based on. I'd like to thank Tufts Create for funding. It's a internal funding that helped me kind of spend a whole semester reading about gardens early on, and that was really important for me actually doing this. The cities of Medford and Somerville, particularly Somerville, who were really engaged and thoughtful and wanted their park's logo on all the signs and who were really down with pollinators. That was really cool to see, and without them, I couldn't have done that last experiment. And also without the community members who welcomed us into their local parks and also their neighborhoods, I wouldn't have been able to gather any of that data. And it was so wonderful to meet all those people. I'd like to thank Sumita, who was my GIS professor in 2019, who, as was really sad about my dissertation work, I was taking this GIS class, and Sumita assigned a class project, and she said, you have to gather some kind of spatial data. And that's when Elizabeth said, what if you gathered something with Google Street View about gardening? And then I did a class project that turned into my entire dissertation. I sent her an email last year saying, thank you, by the way. I'd like to thank Mike Rossi, who kind of helped with so many things that I don't even know if they're his job, but he helped with them anyways, which I really appreciated. And Michael Dwar, who did similar things. Kimberly and Maxime and Doug and any of the other front office staff who I'm forgetting were also incredibly important to the functioning of my life. And I'd like to thank also my undergrad mentors, Helen Rod, who helped me decide to go to grad school when I was really lost at the end of undergrad. She just said, "Do you like research?" And I said, "Yes." And then she said, "Well, then I think you should." Megan Fredrickson, who was my advisor in senior year of that year, who helped me write all the stuff that got me into grad school. And James Thompson, who was my first ecology teacher in undergrad, and who also paid to send me to Rocky Mountain Biolab, where I met my partner. I'd like to thank Avalon, who was just a great friend and always coming out in the field and advising me on stuff. Dylan MacArthur, who I met at Rocky Mountain Biolab and is now a friend in California as well. Ayana, who's a new friend at University of Nevada, who made my poster for this talk. Kevin in the Orions lab, Gabby, and everybody in the Orions lab, Isaac, who was a great friend, and Kelly's Diner for feeding me. I just want to specifically shout out Karen. I love this picture because I look like you're the teacher and I'm the one who's like the student presenting my poster. And that is often how I felt working with you. I feel like I learned so much from you, and you completely, I could not have done like any of this stuff without you. We worked together for like 5 years. It's amazing. I feel so lucky. Thanks to Nick. I really was like, I'm not gonna cry. And you said, yeah, okay. Nick, you've been so important to me. I love you, buddy. I don't know what else to say. I'll say more. I came up with half of the ideas. I came up with half of the ideas for this project in conversation with you, and the other half in conversation with Anita. So, and okay, Elizabeth was a third too. So I guess it's a third. I want to thank everybody in TPI. I can't read that list because it's too long, but you brought so much fun and community to my life. I want to thank my family who hate being in photos, so I picked a photo where they're not turned around. I didn't want to embarrass them. And my sibling who couldn't be here today, but also I love very dearly. I also want to thank the truck. That's my dad's old truck, and I also couldn't have done any of the dissertation without that, I don't think. And then last, I just want to say thank you to Anita, who I met at Rocky Mountain Biolab in this weird picture. This is the earliest photo that exists of us. And I don't know. I couldn't have done anything without you. And I feel like you mean the world to me, and I'm so deeply thankful for everything. And I put in these slides to thank the cats last because I just wanted to say thank you to the cats. It's gingerbread and clementine. Really, you all mean the world to me, so thank you. And I will take any questions.
Well, I think that's a minute to prepare for questions. Before you all leave, I will remind you that we will be having a closed door defense with this committee. So traditionally, the public questions are not asked by committee members. Save them. And then we think at about 2:15 we'll be, exhausted from questioning Atticus, and we will be gathering to hopefully celebrate in the lobby outside the biology office in the SEC. And then at 4 o'clock today, Emily Miner, Atticus's external examiner, who came in person from Chicago, will be giving a talk on her work on landscape ecology of gardens in Chicago in relation to birds. So I hope you can all come and learn more about pollinator gardens. And now we can have about 5 minutes of questions, and I'll cut it off if it seems like it's running too long.
I think I can, yeah, I'll pick. Okay,
yeah.
Helen.
Hey, really, really wonderful talk. Congratulations. It was so lovely to see. I thought it was really awesome work. The question that popped into my mind when you were looking at the nearest neighbors, the 10 nearest neighbors, So I imagine in some of the cities you were looking at, those neighbors were further apart.
Mhm.
And so I'm curious if you think that potentially the social contagion effect could be larger maybe
Mhm.
when the neighbors are closer, like, what thoughts do you have about that?
We had the same thought, which was that nearest neighbor is agnostic to how far away those neighbors are. And so we tried binning, so we tried saying, okay, let's take all the different neighborhoods and bin them by lot sizes. And we had 5 bins from small lots to big lots. And then we drew those contagion curves for all those neighborhoods. And actually, they all look pretty much the same. And the strongest contagion between nearest neighbors, I cannot explain this, we observed in the largest lot neighborhood bin. And then the rest is kind of like noise. I think there's maybe other reasons for that. Like, I think there's certain kinds of people that live on large lots. I think I was assuming that they see their neighbors less, and I don't know that that's actually true. They might talk to their neighbors more than I do. I have no idea. So it was something we tested and we didn't actually see an effect.
Cool.
I think, I don't know. Oh yeah, Nick.
This is a really cool. I love this research. It's really cool. I don't know if you guys are doing this already. It's not really a question about the science per se, but more about the larger project. Do you guys have like a banner or a stamp or something, a logo that you like give people if they have native flowers in their gardens? I know like labels are weird, but people love labels.
Yeah.
And if you like give people, they do this in South Africa with wine farms. If wine farms have native plants in them, then they get the biodiversity certified sticker on
Mhm.
the label. You can sell the wine for more. The wine farmers also like it because it
Mhm.
gets them talking about
Mhm.
themselves. Could you like think of maybe coming up with like something like what Karen designed, giving it to all the people in
Mhm.
their gardens?
Yeah.
And they can like put it on Zillow or whatever,
Mhm.
and it could go back into the
Mhm.
whole initiative.
Awesome. Yeah. So that's a great idea. I'm going to repeat the question because I think the Zoom people can't hear them. I just realized. The question was, have you thought about signage and how that could be valuable, like certification, signage of gardens? A lot of people have programs for that. I've thought about getting involved in them before. TPI, the Tufts Pollinator Initiative, actually did produce a sign at one point, and we sold it to people. I think one of the valuable things there is that it signals intentionality to the people around you. And I think that's really important for a novel practice. Nobody needs a sign explaining what a turf grass lawn is, but when you have a thing that people haven't seen before and are not quite sure what it is, they might read into it things like a lack of care. And that's really important. I think another thing about signage is that it could increase contagion. It could also decrease contagion if people hate it. Like, I don't know. I think it's an interesting question about whether signed gardens are better liked or not. I think there's a perception that they are, but I could also see negative polarization too. So yeah, Gabby.
Okay, this is such a cool study, really fantastic talk. I have a toddler and you're always supposed to give them choices for the illusion of agencies. One, I'll have 2 questions. One is, just if you have kind of any musings about the lack of relationship between lot size and species richness, like maybe some cognitive upper bound on managing multiple species. And the second, which I think you pointed out as a potential future direction, so you thought about like more complicated interactions between any of those random effects like income and kind of susceptibility, I guess, to social contagion, like whether certain things are more contagious.
Yeah, so I'll address the second question first, which is, I think it's a great question, like, because my main goal was to try to estimate away neighborhood effects just to get a contagion, but I think neighborhood effects could clearly affect contagion, like different kinds of people live in different places and they could be more susceptible or less susceptible, right, to contagion, but also to just the idea of gardening in general. I think to get at that, I might want to compare between cities, like between whole metro areas, because I don't know, that might be an easier way, because then we don't have to try to do the interactions. We could just say like, you need a lot of cities really, but getting at the causality would be really hard too. What was the first question again? Oh, lot size not having an effect on species richness. I think it's funny. I thought it was really surprising and interesting, and it made me think, wow, these like front yards are under such strange conditions that they violate universal laws of ecology. And I think that's so cool. They're under such strong human management that and it's true of weeds too, like the unintentional plantings. So it's not just like I think it's because management matters so much that that's all that matters. And if management isn't stratified across lot types, like how often you mow, for instance, then, I don't know. So I thought that was really cool. Somebody, and one of my reviewers was like, of course, basic ecology doesn't apply to the city. And I was like, I did not assume that. So I don't know. I think it's neat. I think you were.
Yeah, just on the income side of things, maybe two related questions. One is, you could imagine some municipal government thinking that you could actually help solve that problem and incentivize people to, financially incentivize people to create pollinator habitats. So I just wonder whether your work combined with everything else would say that that's true or maybe not. I mean, I know that you mentioned sort of the income and education contagion, and that might be an issue. And then the related question is, if we think that there's an income aspect, I just wonder whether you have a sense of sort of what actually, it's probably on the cost side, presumably, right? So do we think it's setup or maintenance? Because like, again, from a policy point of view, you could imagine two different ways of thinking about that.
Yeah.
Give a grant, or maybe you're not going to support people here.
Totally. So the second question was, what is the relationship between, so why is income limiting to people gardening potentially? I think there's a lot of possible reasons. I think cost is often reported. I mean, they've done surveys, and native plants and pollinator friendly plants are expensive. I think my theory is that it's more of a perception of cost issue that's an even bigger problem. I think people don't acknowledge the money they already spend on their lawns because they perceive it as being inevitable. And they don't correctly, at least I found myself having to remind myself of the cost of the lawn care industry, which is a multi-billion-dollar industry every year. And so it is not that lawns are free. They are extremely expensive and costly to maintain if you want a good lawn. I think it's an unknown question whether maintenance and stuff is actually more expensive, but it is perceived as more expensive. And I think actually, I would say that probably a bigger reason for the income effect is that it is trendy in certain types of communities and certain types of people. And this was an idea that Caleb actually started convincing me of, which is that who lives in high income, small lot size neighborhoods in 2021, which is when this data was gathered. In our area, it's increasingly like a certain subset of like liberal yuppies. And those people tend to be the ones who are pollinator gardening right now for a whole lot of reasons, including social contagion, like their friends are doing it and they're friends with people who are also yuppies, but also factors like, that's what, I don't know, sociology. Catherine.
Under monarch data, so you analyzed it as number of eggs per leaf or per area, and so that caused you to have this saturation effect. And I'm wondering if you ever analyze your data just as total number of monarch eggs, because that would then, maybe tell you whether you had a positive effect of just like more eggs because of the garden.
That is a good question. I don't know that I ever actually ran the analyses of that. Yeah, I don't think I did run those analyses. I think we set it up in the beginning about rates of use, but there was certainly variation in the total number. If I ran it, it was not that interesting, and that's why I don't remember it. But it's also possible I never asked. There was big variation. Some parks had, every park got some eggs, which was cool. But I actually have a slide for this. Beware, hidden backup slides ahead. Hold on. There's a map, I promise. Don't worry about any of this. Okay. So we actually saw pretty big variation in the total number of eggs. One park had 8 eggs, and another park had 147 eggs. And 147 eggs, anything over 100 is like kind of insane because that's they're this big, the pots. So it's really neat that you can put out a pot that big and get 100 eggs if you're lucky enough to be in a neighborhood that got 100 eggs. Eric.
So you started a little bit, but I want to ask a follow-up. You can speculate a little bit about how general these results might be, particularly, comparing sort of old Northeastern, relatively liberal around the country,
Yeah.
how can these things generalize?
Mhm. It's, so the question, sorry, I've been bad at repeating the questions, and maybe I should see if the Zoom even has any questions. So the question was, how generalizable are these trends? And like to other cities and other situations? I think the answer is I don't know. And I think one thing I would say is I don't think the method is completely plug and play. I think that if you were, I think it would, you could totally do Google Street View in any city on earth to look at front yards, and I encourage people to do that. But I would say you do need to go visit in person and establish your own criteria. Like you need to train people on how to identify images of that region, because gardens look completely different in Arizona to here. Both aesthetically, what kinds of plants they are. The images are blurry. You can't actually see. It's more of a gestalt, right? And so you also need to visit in person and see, like, for the type of gardens and the type of imagery we have in this region, does it actually align? Like, is there actually gardens there? But with ground truthing, I think you could apply it to any city, and with training, you need to learn about that. My answer is, I think this is a pretty good region for pollinator gardening, and I'm glad I studied it here, because I think it's in a great stage to study social contagion. It is rare, but it is not so rare that we only found 10 gardens. We found 2,000 gardens, which is only 2%, but it's also enough. And I think that's also a stage at which you can see contagion operating, right? It's not like completely dominant yet. I think other regions also, the upper Midwest is, I think, the capital of pollinator gardening in the country. So Minnesota would have been another good region, perhaps. I think they have more even. There's a prairie gardening movement there. And then California has a whole different thing going on. Actually, xeriscaping throughout the West, so low water landscaping also has biodiversity benefits probably. And I think also would be identifiable on Google Street View, just a different project. Okay.
This might be a good time
think this is it.
Atticus again.
Thank you, everybody. Yeah, thank you, everybody on Zoom.